Okay, so a little introduction, take a break, do a bit more stuff. So what we're going to do today is look at how you can tell the structure of a crystal using X-rays. So how X-ray diffraction kind of works. So this is the theory behind it. There's a bit in the middle of this lecture that gets a little bit heavy that I'll sort of go through relatively quickly. I can go over with people that are interested. But the end results, that there's a couple of sort of main results that drop out at the end that are actually important. So the derivation in the centre is just there to scare you for no other reason than that. I think it's instructive, but don't panic if it goes a little, because it does use a few of these quantum ideas that have just come in from left field that we've not fully installed into your conscience yet. So what is the idea? I think I said it a little while back, that if we have a crystalline material, by definition we've got these periodically repeating positions of atoms, and from our understanding of what atoms are, or even if you haven't got that, because the original diffraction experiments were suggested before the atomic structure of atoms was properly confirmed, at least there's something varying periodically, so it should act as a sort of diffraction grating, so we should be able to shine light of an appropriate wavelength or frequency through it. It should act like an interferometer, like a diffraction grating in the experiment you've done, so you should get a bunch of maxima and minima, and by an appropriate analysis of the maximum and minimum positions, you should be able to determine the width of your diffraction grating. And basically the width of the diffraction grating is going to be our lattice constant. That's what's going to emerge from that. If you go a little bit further, which we won't really do in this course, I'll mention it, but depending on the positions of the atoms within the unit cell, then the maximum and minima intensities get further modulated, and you can, in principle, detect exactly where all the atoms are in the cell. So this is sort of incredibly powerful. It's used to reveal the structure of DNA. You see the cubes downstairs with some scientists that did it. So yeah, so diffraction grating. We now know that the spacing of atoms is of that order. There were estimates from sort of volumes, densities, what happens when you solidify stuff, what that would be, and the suggestions were to shine X-rays at this stuff and see what was absorbed. So basically, what's going to be done in a simple picture is this. But life gets considerably more complicated, because a real crystal isn't just a strip with this one set of diffraction gratings, but it's got all the different orientations of the crystal that you can think of. Each orientation, each sort of repeating set of lattice planes in the crystal can act as a grating, so you get a sort of superposition of all these different grating effects present. And so what we are going to predict at the end of this is, given the structure of a crystal, we are going to be able to predict what angles you would expect the maxima and minima, well, basically of the maxima, which angles we would expect that to be at. And then if you perform an experiment, you make a measurement, and you can do a little bit of analysis that I'll talk you through. We'll do together in the second half for crystal. You can then sort of work backwards and match the peaks in the diffraction pattern. It's not really a spectrum because there's no variation in the frequency of the light. But in the diffraction pattern, you can match it to what you expect from the theory for a given model, and where they match, you can say, this is my crystal. So what we did at the end of last week, I think it was the end of last week, we did it last week anyway, was this expression, so that the distance between the different planes as you turned the crystal round, that had a sort of different orientation pointing upwards, and then you looked at the sort of repeating pattern going along that direction. We said that they would be identified with a distance, so the spacing between the planes in that orientation would be given by 2 pi, the length of the reciprocal lattice vector, which is going along that direction. So these Miller indices are a way of indicating those directions, and they're these integers that tell you how many of these reciprocal lattice vectors that you put into this given direction that you've chosen. And from the geometry, from the way it's decided, you end up with something that's basically a normal vector to that direction. So essentially it will be the phi vector will be a vector in this direction, and its length will be inversely proportional to that thing there. So I've got a suspicion I'm going to run out of battery if I really go into this in detail here now before that, so I'm going to go and get my charger. I'll take a break, a quick break now. I'll let you sort of flick ahead if you like.